Dear viewers, this video is part 2 in the 4-part series on infections overview in the Medicine for Anyone in the YouTube channel. The eukaryotic cell. There are two features in the single eukaryotic cell with a particular bearing on infection and immunity. They are both concerned with the movement of information inwards and outwards, particularly important in maintaining the stability of a complex multicellular organism. One is the process by which the cell learns about its environment by using a series of receptors on the outside surface of the cell membrane, which are specialized for recognizing the surfaces of other cells, including pathogens, as well as hormones and other chemical messenger molecules with an influence on the cell. The other is the process by which the cell conveys information from its own interior to other cells. This might include the fact that it has become infected with, for example, a virus, and is performed by specialized MHC, that is, major histocompatibility complex molecules, that transport bits of protein from inside the cell to the surface. The left side figure is a normal eukaryotic cell, has a cell membrane, mitochondrion, cytoplasm, chromosomes packed within a well-defined nuclear membrane, lysosomes, endoplasmic reticulum, and nucleolus. The right side figure is a virus-infected cell. It shows receptors for chemical signals and for virus particles, as well as MHC virus complexes. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Coming back to the discussion, bacteria cannot be considered primitive. Indeed, it is a mistake to call any species of creature alive today primitive because it has been around for a great deal longer than we have, sometimes in extraordinary conditions. For example, there are bacteria that can survive one freezing. Psychrophiles are a type of organisms that survive in extremely cold temperature of 15 degrees centigrade or lower. Examples of psychrophiles such as Chrysobacterium greenlandensis can be found in glaciers, deep ocean waters, and polar ice. 2. Boiling. Pyrococcus furiosus, the furious fireball. It was isolated 30 years ago from a shallow submarine hot spring on a beach in Volcano Island by the father of high temperature microbiology, Professor Carl Stetter. It grows best at 100 degrees centigrade, the boiling point of water. 3. Enormous doses of radiation. The bacterium Deinococcus radiodurans shows remarkable resistance to a range of damage caused by ionizing radiation, desiccation, UV radiation, oxidizing agents, and electrophilic mutagens. 4. Total lack of oxygen. Anaerobic bacteria or germs that can survive and grow where there is no oxygen. For example, it can thrive in human tissue that is injured and does not have oxygen rich blood flowing to it. Infections like tetanus and gangrene are caused by anaerobic bacteria. 5. High salt concentrations. Microorganisms called halophiles, that is salt loving, actually require high salt concentrations for growth. These organisms are found in marine environments where salt concentrations hover at 3.5%. Extreme halophilic microorganisms such as the red alga Dunaliella salina, and the archaeal species Halobacterium. All these extreme conditions would finish us off in no time. Clearly, bacteria are great survivors, and by no means they are not about to become extinct. Useful bacteria. If bacteria were to disappear, life on Earth would soon suffer. To take just two examples, bacteria in the root nodules of plants fix nitrogen in the soil, essential for healthy plant growth. Note, Rhizobium is a genus of bacteria associated with the formation of root nodules on plants. These bacteria live in symbiosis with legumes. They take in nitrogen from the atmosphere and pass it on to the plant, allowing it to grow in soil low in nitrogen. The soil air content or air filled porosity can vary between approximately 30 and 60%. The billions of bacteria in the rumen of cattle are needed to digest the cellulose of the grass they eat. Note, Prevotilla, Buteri vibrio, and Ruminococcus were the most dominant bacteria in the rumen. The rumen is the largest stomach compartment consisting of several sacs. A cow's rumen functions like a large food processor. In fact, millions of bacteria naturally live in the rumen and help the cow by breaking down plant parts that cannot be digested otherwise. These tiny organisms then release nutrients into the rumen. Even in non ruminants like ourselves, the bacteria peacefully resident in the intestine appear to be beneficial. When pathogenic bacteria and fungi may replace them, as when antibiotics kill the normal resident bacteria in the intestine, it may lead to digestive problems. These friendly bacteria are sometimes called probiotic, that is, in favor of life. And we should not forget the useful jobs that we have invented for bacteria, fermenting milk into cheese, recycling waste, and in the industrial synthesis of enzymes and antibiotics. Only some bacteria cause human disease. Only very few of the thousands of species of bacteria are pathogenic. Most are not even parasitic. Of the roughly 400 recognized genera, groups of species of bacteria, less than 40 contain species that regularly cause disease in humans, although there are others that cause disease in other animals. On the whole, different species of animals have different species of pathogens. But there are some bacteria that can cross the species barrier from animal to human under conditions of close contact, such as plague from rats, brucellosis from goats, and in the days before pasteurization of milk, TB from cattle. The so called zoonoses are often particularly serious and even life threatening to us because the rats, cattle, etc. are the natural hosts with which the bacteria have come to reasonably peaceful terms over the millennia, whereas we, as occasional hosts, have not had this advantage. The overall view of microbes residing in humans. Perhaps it is not surprising that a general understanding of the microbiota, the overall microbial inhabitants of our body, is essential in the evaluation of infectious diseases. Microorganisms occupy almost all imaginable ecological niches. Microbes have been isolated from deep sea sites where they survive very high pressure, from extremely cold and extremely hot regions where hyperthermophiles grow optimally at temperatures well in excess of 100 degrees centigrade, and even from rocks where they can exploit chemical substrates for energy generation. It is no wonder then that microorganisms should also exploit other living organisms as potential habitats, from viruses that use other microorganisms as hosts, example bacteriophages, through to microorganisms that occupy various ecological niches within and upon the human body, sometimes to the benefit and sometimes to the severe detriment of the host. Microorganisms have been supremely successful in evolutionary terms, and they contain enormous untapped reserves of biodiversity, much of which is to be found in those that we can neither isolate nor grow, and which make up most microbes on the planet. Since microorganisms reproduce much more rapidly than their mammalian hosts and have several specialized mechanisms for horizontal gene transfer, it is not surprising that they are often able to evolve quickly to stay one step ahead of any mechanisms that exist or are invented by humans to control them. Note: Escherichia coli bacteria doubling time 20 minutes, viral replication time about 8 hours. The control of infectious diseases over the last century or so has been a major achievement, relying mainly on improved public health systems and social conditions, as well as on technological advances such as antimicrobial drugs and vaccines. Hope of eradication of microbes afflicting humans. Optimism about winning the battle with infectious diseases was bolstered by the eradication of smallpox in 1977, achieved by a monumental worldwide public health and vaccination program. The sense of complacency, if at all it was there, was shattered soon afterwards by the advent of the global catastrophe that is AIDS, along with the resurgence of ancient killers such as tuberculosis and the emergence of apparently new or re emergent threats such as bovine spongiform encephalopathy (BSE) and West Nile fever. Note: BSE, so called mad cow disease, is short for bovine spongiform encephalopathy, which tells us what the pathology is: spongy areas of degeneration in the brain of cattle. BSE is very much like scrapie, a long established disease of sheep, while CJD is reminiscent of Kuru, a condition restricted to New Guinea and shown to be spread by the unusual, that is to us, habit of eating the brains of dead relatives. West Nile fever is a single-stranded RNA virus that causes West Nile fever. It is a member of the family Flaviviridae from the genus Flavivirus, which also contains the Zika virus, dengue virus, and yellow fever virus. The virus is primarily transmitted by mosquitoes, mostly species of Culex. In 2009, the world found itself dealing with the long predicted global pandemic of influenza, which severely stretched and tested the ingenuity and organizational abilities of human society in its attempts to control the impact of the virus. That this pandemic had a relatively minor impact, despite rapid and widespread dissemination and transmission, was largely due to the generally mild nature of the disease caused by this virus. Lessons for public health regarding control are still being learned from this incident. Far more devastating, at least locally, has been the Ebola epidemic in three countries in West Africa between 2014 and 2016, a catastrophe that has had far reaching consequences on health service infrastructure in those countries and on international responses to epidemic disease. Note: Ebola virus disease (EVD) is animal borne, with bats or non human primates being the most likely source. Infected animals carrying the virus can transmit it to other animals like apes, monkeys, and humans. It causes explosive epidemics of the highly lethal Ebola virus disease, the largest ever being in 2014 to 16 in West Africa when it claimed over 11,000 victims. The virus punches holes in capillaries and blood, teeming with viruses, oozes into the tissues and body fluids. So, while the patient is prostrate with high fever, severe pain, generalized bleeding, and catastrophic vomiting and diarrhea, the viruses in body fluids take the opportunity to pass to unsuspecting family members and hospital staff. New disease threats will continue to emerge, leading to consequences that are difficult to predict, such as the emergence of Zika virus as a recent example. Note: Zika virus primarily infects Asian and African primates, but can also spread among humans, began to travel westwards across the Pacific Ocean in 2007, reaching Brazil in 2013. In 2015, it caused a huge epidemic of a mild flu-like illness in South America, with serious consequences in pregnant women because of spread to the unborn child. Several birth defects have been reported, most commonly microcephaly, meaning small head. The virus has now increased its vector range, that is, host range, to non tropical species and so is set to spread to North America and Europe. So, dear viewers, this is the end of video part 2 in the 4-part series on infections overview in the Medicine for Anyone in the YouTube channel. Thank you for watching.